Hello and welcome back. This is the second of two lectures where we explore the two main types of virus infections in a host. Remember, last time we talked about acute infections, the ones that come and go, and today we're going to consider persistent infections, those that last the lifetime of the host. Acute infection, to remind you, is rapid and self-limiting, whereas a persistent infection will last the life of the host. Every virus has its own type of persistent infection, and as I said last time, most persistent infections probably begin as an acute infection where the symptoms may come and go, but the virus remains. Here's a graph showing you the two different kinds of infection, the acute infections that we talked about last time, where we have a period of virus production, the blue line, which is on the y-axis, and the red bar is disease. So acute infection is a very defined period of virus production and disease. X-axis is time, of course. The other segments of this graph show you the different kinds of persistent infections that we recognize, and we call some of them latent. These are particularly special persistent infections that we see with the herpesviruses, and we'll talk about these today. These are characterized by bursts of virus production with or without disease. Some persistent infections have no disease. They're asymptomatic, and virus is produced for the lifetime of the host. An example of this would be HCMV infection of rodents, or a polyomavirus, JC virus infection of humans, where we have continuous virus production with no disease. And then we have persistent infections where there's an initial acute infection, followed by long-term production of virus, and eventually death. And that's HIV, HTLV, and Measles, SSPE. And we'll talk about measles today and HIV towards the end of the course. Persistent infections in general occur when the first infection, the first primary infection, the acute infection, is not cleared by the immune response. And again, that is an infection that lasts the life of the host. Viruses, proteins, genomes continue to be produced. Not all of them for every virus. We'll see each is slightly different. But the viral genome is always there. Sometimes there are no viral proteins made whatsoever. And that's part of the strategy for evading the host immune response. And these are some of the viruses that we're going to talk about today as examples of persistent infections. We're going to talk about measles virus, polyomavirus, hepatitis B and C viruses, and herpes viruses. There's no single mechanism that is common to all viruses that cause persistent infections. There are slight differences between each virus. But in general, what we can say is when the viruses are not cytopathic and when the host defenses are reduced or interfered with, this is a recipe for a persistent infection. All of these viruses have to encode gene products that in some way modulate the immune response. Otherwise, the infections would be cleared. Here's a table showing you some known persistent human infection. On the left, the virus. In the middle, the places in the host where persistence occurs, the tissues, and the consequences of that infection. And the ones with a red asterisk we are going to discuss today, so we're going to go over quite a few of them, but there are others as well, HIV in particular, which we'll discuss on another time at the end of this course. You can see, for example, Epstein-Barr virus persists in B cells and the nasopharyngeal epithelium. The consequences, Burkitt's lymphoma, Hodgkin disease, certain kinds of cancers. For some of these viruses, there is no apparent consequence of persistence. The virus is there for the life of the host. An important component of all persistent infections is modulation of the immune response. The reason why acute virus infections are cleared is because there is minimal antagonism of host defenses. These viruses that cause persistent infections effectively modulate immune defenses, and a main intervention is at the cytotoxic T lymphocyte response. Just to remind you, we have an infected cell which is displaying in MHC class 1 peptides, shown by this orange box, which are derived from viral proteins that have been processed intracellularly. We talked about this a little while ago. Cytotoxic T lymphocytes, CD8 positive T cells, recognize the infected cell by virtue of this peptide presented in class 1, and they initiate a killing response of the infected cell. And this is a response that is antagonized in many persistent virus infections. Here's an overview of the MHC1 system and modulation by different viral gene products. And if we go all the way to the end, we have MHC class 1 with a viral peptide in it. It is being recognized by a CTL to initiate killing. But this display of a peptide in class 1 begins all the way in the nucleus, of course, with transcription of MHC class 1 genes. And that process is antagonized by viral proteins. So all these yellow boxes show you viral proteins that are antagonizing the red bar at different steps of MHC1 antigen presentation. We have some viral proteins that interfere with export of the mRNAs from the nucleus. We have gene products that interfere with the proteasome itself, transport of peptides into the endoplasmic reticulum. We have viral gene products that actually stimulate the translocation of class 1 from the ER back into the cytoplasm, where it's then degraded. It's not where it should be. It should be shipped up to the plasma membrane via the Golgi. Viral proteins that interfere with transport from ER to Golgi. And we have viral proteins that actually cause the cell surface MHC1 to be internalized and degraded. So all these viruses are causing persistent infections, and they all interfere at some point with MHC1 presentation because this is an important mechanism for eliminating virus-infected cells. Some other comments on escape. Another one that we haven't talked about before is the generation of mutants that escape recognition by CTLs. Here on the right again is a summary of a CTL killing infected cells. The key is recognition of a viral peptide by the T cell receptor in the context of MHC type 1. That process is detailed on the left. Viral peptide shown in green, and MHC class 1 is the gray molecule shown here. We have nice recognition of the peptide by MHC. You see, not many amino acids are involved here. And if we change one of these amino acids, we can abrogate recognition of the peptide by the class 1 molecule. Viruses that produce altered proteins as a consequence of mutation, they won't be recognized by CTLs because the peptides won't be displayed in class 1. Those viruses will be selected for, and they will predominate. And those infected cells won't be eliminated by CTLs because the peptides don't fit into the MHC class 1. These changes can work at two levels. They can prevent binding of the peptide to MHC class 1, or they can interfere with the T cell receptor recognizing the bound peptide, as you can see on the bottom here. So normally this T cell receptor would recognize the peptide in the context of class 1, but sometimes the mutation shown by the blue box will prevent recognition by the T cell receptor. And in addition, changes in the viral proteins can also affect their processing from precursors into the peptides. So CTL escape mutants can work at three levels. They can affect binding of the peptide to class 1. They can affect recognition of the peptide by the T cell receptor, and they can affect processing of the protein by the proteasome. Another aspect of interfering with CTL function is that the CTL may die. So in some cases, when a CTL engages an infected cell, the CTL dies instead of the infected cell. This is an unexpected turn of events, of course, but it's another amazing example of a viral counter-offense to host defenses. Here's how it works. Activated T cells have a membrane receptor on their surfaces called FAS. and this binds a membrane protein called Fas ligand or Fas-L. When Fas on activated T cells binds Fas ligand on target cells, the receptor trimerizes. That triggers a signal transduction cascade, and it turns on apoptosis or programmed cell death of the T cell. In other words, if viral proteins increase the concentration of Fas ligand on the cell surface, any cell that engages it will undergo cell suicide. And there are HIV proteins, there are HTLV proteins, and cytomegalovirus proteins that all appear to increase the synthesis of Fas ligand within infected cells. So when T cells recognize those cells, they're killed. The T cells are killed and not the infected cell. This is an insidious mechanism. It happens to be co-opted from a host process that would normally limit immunopathology. Normal Fas-mediated CTL killing removes activated T cells when they're not needed after an infection, or if their presence in a tissue may be detrimental. And certain organs or tissues that are irreplaceable, like those in the eye, are free of potentially destructive T cells because those cells in the eye have a high concentration of Fas ligand on their cell surfaces. So this is a normal process to limit immunopathology that has been co-opted by certain viruses to prevent killing of the infected cell. And as I just mentioned, a number of organs within us have reduced immune surveillance. And these include the central nervous system, the vitreous humor of the eye, inside the eye here, and areas of lymphoid drainage, devoid of initiators and effectors of the immune response. We don't want vigorous CTL killing in these areas, and therefore we modulate it. We reduce immune surveillance. And I just mentioned that the eye cells have high Fas ligand on their surfaces, so that if there are any activated T cells that come in, they'll be killed by apoptosis. We don't want vigorous immune responses in these tissues because they can be damaged by the inflammatory process that would result. Fluid accumulation, swelling, ionic imbalances, not good things to happen in the eye and in the brain. As a consequence of these diminished responses, persistent infections of these tissues are common. Not every cell and organ has the same immune defense, and that can play a role in persistence. Not too many years ago, after the Ebola virus outbreak in Western Africa, we began to recognize that the virus could enter the eye and persist there. And here's an article in the New England Journal of Medicine in which it was reported that the virus could persist in ocular fluid. This was a patient who recovered from Ebola virus disease, seemed to be fine, but of course he was monitored periodically, and he was found to have severe unilateral uveitis. They detected infectious virus in the aqueous humor, the fluid of the eye, 14 weeks after the onset of his disease and 9 weeks after there was no more virus in his blood. And he was assumed to be free of virus, but in fact the virus had gone into his eye and persisted there because it was not cleared. Remember, the eye is an immune privileged site for reasons that we just described. This has now subsequently been described in testes as well, another immune privileged site. And individuals who recover from Ebola virus disease can have virus persisting in that site as well. Some viruses can infect immune cells. For example, Measles virus infects antigen-presenting cells. HIV infects a variety of immune cells. And this, of course, is a mechanism of persistence, because if you interfere with immune defenses by compromising the function of immune cells, by infecting them, that can lead to virus not being cleared. And so another way that persistence can occur. Our first question is, which of the following are features of persistent infections? They last the lifetime of the host. Viral immune modulation is involved. Immune cells may be infected. They may occur in areas of reduced immune surveillance, or all of the above. If you want to pause the video at this point, because I'm going to give you the answer to this question. The answer, of course, is all of the above. They last the lifetime of the host. Immune modulation is involved. Immune cells may be infected. They can occur in areas of reduced immune surveillance. All of these things I've just told you. So the answer is E. Let's now look at a series of examples of persistent infections so that you can see the different mechanisms that are involved. And this one may surprise you, measles virus, because last time we talked about it as a typical acute infection. And it is. It's a classic acute virus infection. Remember, it's a very contagious human virus spread by respiratory routes. Globally in 2014, 115,000 deaths, all of which are preventable because the vaccine works. And typically, you get infected with measles, you have a period of disease, you clear the infection, and you have lifelong immunity. So where does the persistence come in? It comes in this disease called SSPE, which I mentioned last time, subacute sclerosing panencephalitis, which is a progressive degenerative encephalitis that occurs many years after the initial measles virus infection. It happens about 1 in a million cases of measles. 6 to 8 years later, the child begins to show signs of neurologic degradation and eventually dies. These are 100% fatal. And we're not quite sure how this works. We know that there are nuclear protein particles in the brain, but there is no infectious virus, and viral genomes appear to spread synaptically between neurons. But why it persists and in what form and the pathogenesis, why the degeneration occurs in the first place, is not understood. So that's an interesting example of how a fraction of acute infections can eventually become persistent. Another example is polyomavirus. These are small, double-stranded circular DNA-containing viruses. We talked extensively about SV40, which is not a human virus, but it is a polyomavirus, a member of this family. We talked about it in terms of its being used to study, for example, transcription and DNA replication. But there are some polyomaviruses that infect humans, and they're shown on this figure by the red asterisk. And so there are polyomaviruses that infect many different kinds of animals. They're classified into Wookie polyomaviruses, avi polyomaviruses, and ortho polyomaviruses. The Wookie polyomaviruses are so called because two members are named WU and KI. WU stands for Washington University, where it was isolated, and KI, the Karolinska Institute in Sweden, where it was isolated. Someone with a nice sense of humor decided to call these the Wookie polyomaviruses. And they don't infect Wookiees, actually. They infect humans. You can tell by human polyomaviruses. But there are others that infect humans as well, like BK and JC. And these are viruses that seem to infect us early on and stay with us for our lifetime. If you look at serological studies in human populations, a good fraction of the human populations seem to be infected, and were infected for life. These viruses infect a variety of organs: kidney, intestine, respiratory tract. And you can shed as much as 100,000 particles per mil in your urine with no apparent disease in the kidney. So these viruses are replicating in the kidney, they're shed in the urine, they're replicating in the intestine, they're shed in feces, they're replicating in the respiratory tract, and they're shed in your breath. You seem to be fine, and the people who contract it. I always like to say, next time you go in a bathroom, just think about this, because when toilets or urinals are flushed, they generate aerosols. So you are inhaling probably polyomaviruses. We don't know how these viruses persist. But again, most people seem to be infected, and there doesn't seem to be any pathogenic consequences, except if you are immunosuppressed. For example, if you have multiple sclerosis, one of the treatments is giving you a monoclonal antibody that is immunosuppressive. And in a fraction of those patients, that can result in these viruses replicating in the brain and causing disease. So immunosuppression leads to pathogenesis, whereas in an immunocompetent host, there is no disease. And the disease that results in this case is called progressive multifocal leukoencephalopathy, PML. So this is a specific brain disease caused by replication of a polyomavirus in patients who are immunosuppressed. If you want to learn more about these viruses from a person who has done research on them his life, Bob Garcia, listen to TWIV 250 on Wookiee viruses. Now, let's talk about some viruses that persistently infect the liver. And one of them is Hepatitis B virus, which we've talked about before because it has that interesting double-stranded DNA genome with a gap and a protein and an RNA linked to it. And these are viruses that are enveloped. And remember, they have reverse transcriptase in their life cycle. These viruses are transmitted among humans by exposure to blood. And that can happen during childbirth. If you get a transfusion with contaminated blood, it can happen during sex, drug use, intravenous drug use, reusing contaminated needles, which happens often, tattooing using improperly sterilized needles, and you can get them in healthcare settings as well. The main target of this virus is the hepatocyte, a liver cell. Most of these infections are resolved if you're an adult. 95% of adults resolve the infection. If it's an acute infection, it's gone. But if you are a newborn and you're infected, which can happen, so if your mother is persistently infected, you can receive the virus at birth. And because they don't have a good immune system, they don't resolve the infection, it can become persistent. This is a map showing you the current status of infection with hepatitis B virus globally. This is from the World Health Organization. 257 million people are infected globally, and you can see there are certain parts of the world that have a higher burden of infection than others. The most infected area is the Western Pacific, where 115 million people are infected. And this is a combination of acute and chronically infected people who are infected but don't resolve the infection. And here's two graphs that contrast the two outcomes: acute infection and chronic infection. So in an acute infection, you are infected with virus by whatever route, and we're looking at weeks after exposure. We're going a long time here, 100 weeks, and a variety of measurements on the y-axis. So first we can see green, hepatitis B virus DNA rises to a peak and then declines, and after a year, we can't find any more hepatitis B DNA. So this is a classic acute infection where the virus infects and eventually it's cleared. It takes a while, but it's gone. And then we can measure viral proteins. That's in red. Hepatitis B surface antigen is a viral protein that's comprising this outer shell of the particle. You can see that peaks in about 8 weeks or so, and then that declines. You have some liver enzymes in the blood, alanine transaminase, ALT, that also peaks at about 12 weeks. That's a sign of liver damage. Remember, these are enzymes that are present inside of liver cells, and they should not be in the blood. And if you have hepatitis, they're in the blood. We also have IgM antibodies, which rise first. Total antibodies against hepatitis B are very high, and that includes IgG, of course. And you have a lifetime immunity now because you have antibodies against viral proteins. So that's an acute infection. You have symptoms for a defined period of time, then the virus is gone. But as I said, some adults and many infants do not clear infection. They have the initial exposure. You have viral DNA produced, but it never goes away. You see, it keeps on being present past 100 weeks. The liver enzymes continue to be elevated, and the viral proteins are elevated. You even continue to make antibodies, but you do not resolve the infection. Now, the symptoms, the initial symptoms of the acute infections are evident in a defined period of time, but the virus remains throughout the life of the host. And that is without the initial acute symptoms of infectious hepatitis, but you can see the liver enzymes are still elevated, reflecting the fact that there's continuous damage of liver cells. Even though you may not have symptoms of a chronic infection, you certainly have signs of it. Interestingly, the virus does not kill hepatocytes. It is the cytotoxic T cells that kill infected hepatocytes. So if you think about it, there are years and years of continuous production of hepatitis B viruses in the infected liver. They are not cleared. CTLs are continuously clearing them, but they're unable to clear the infection completely as it would in an acutely infected person. We think that exhaustion of the T cells, which is a mechanism by which they're shut down, this prevents them from killing cells. But a fraction of the T cells are still killing liver cells. They're just not able to clear the infection. And eventually you have cycles of T cell killing hepatocytes, regeneration, and killing. This leads to fibrosis and eventually to cirrhosis and liver failure after many years. And in the worst cases, hepatocellular carcinoma, HCC. Liver cancer will develop after 20 to 30 years of chronic infection. And as I said, this is often asymptomatic. You don't have any symptoms. But again, the virus is there, your liver cells are being destroyed, ALT is in the blood, and after all of these years and years of destruction, that leads to cancer. So you can see this is a big problem, especially with so many people infected. Another virus that targets liver cells is hepatitis C virus. It's a completely different virus family. This is a flavivirus, a plus-strand RNA virus with an envelope. This is also transmitted by exposure to contaminated blood during sex, drug use, tattooing during birth. 71 million people infected globally. Here's a World Health Organization map of the number of infections. 71 million globally. And again, you can see that certain parts of the world have a higher burden of infection than others. And many of these infections are transmitted by intravenous drug use using contaminated needles or by the medical reuse of contaminated needles. Here's the pathogenesis of infection. So you acquire infections by the routes that we just discussed. You have an acute infection. You have elevated liver enzymes, hepatitis. You can have jaundice evident in yellowing of the whites of the eyes and the skin. Within 3 to 6 months, 10 to 40% of the infected individuals will recover. They will clear the infection. But 60 to 90% develop a persistent infection that's characterized by depression, cryoglobulonemic vasculitis, porphyria cutanea tarda, and they can go on for the life of the host. In about 30 years, 70 to 98% of the individuals have a persistent asymptomatic infection, and they can live the rest of their lives with this infection. But 2 to 30% develop end-stage liver disease. In other words, they die, and during this persistent infection, you can have jaundice, encephalopathies, esophageal varices, cirrhosis, hepatocellular carcinoma, and the accumulation of fluid in the GI area, ascites. So again, a serious disease in a fraction of the individuals. This virus persists via a number of immune modulation mechanisms, many of them targeting at reducing interferon responses. At the bottom is a graph of the course of a acute and persistent infection, and we have time on the x-axis, months after infection, so we're looking at a year in this particular study. On the left, Y is viral RNA, and this is in the blood, and on the right is a liver enzyme, ALT again. In red, these are cases where the virus is cleared. You have an initial exposure, you have high levels of viral RNA, that's the red solid line. You have liver enzymes rising to high levels, but eventually that's all cleared. LLOD is lower limit of detection. In some individuals, though, as we just mentioned, you have persistence. The blue line, viral RNA persists for here a year at high levels, and the liver enzymes persist at high levels. Again, the virus can persist in the liver, causing damage, and in some individuals eventually liver cancer. So that's why Hep B and Hep C are big problems, because not only do they stay around for a long time and the potential of infecting others is there, but they can lead to death by hepatitis and liver cancer. A more recent problem is that there has been an increase of hepatitis C virus infections in the US since about 2004, and this seems to be associated with opioid injection. This is a slide from the Centers for Disease Control. They're looking here at hepatitis C virus infections in orange. Y-axis for that is on the right. So we're looking at the rate of acute hepatitis C. As you can see, it's gone up 300% in this time period. And at the same time, admissions for opioid injection are also increasing. And the idea is that, and this is particularly among white Americans, the idea is that the reuse of contaminated needles to inject opioids is leading to an increase in infection. And this is not good because a good fraction of these people will be infected for life and may die of liver cancer. Our next question is, what are shared features of persistent infections with polyomavirus, hepatitis B virus, and hepatitis C virus? Genomes are present but not expressed. Liver damage, kidney damage, virus particles are produced, or all of the above. I'll give you the answer now, so if you'd like to think about this, pause the video. The answer is virus particles are produced. In all cases, of course, genomes are present, but they are expressed. I talked about polyomavirus production, HBV, HCV virus production. Liver damage is for HBV and HCV, but not polyomavirus. Kidney damage could occur with polyomavirus, but not with Hep B and C. But they all make virus particles, so that's why that's the answer. Let's now turn to discussing latent infections. And this is a name for persistent infections that has historically been given to the persistent infections caused by herpesviruses. And here's some general properties. And we're going to talk about a number of different herpesviruses today. One is that the viral gene products that allow for a lytic replicative cycle are not made, or they're found in low concentrations. And cells that have genomes in them and that may be making proteins are poorly recognized by the immune system, so they're not eliminated. And of course, the genome persists intact, and that's important. Intact means so that can give rise to new infectious particles, which is what you need to spread the infection to new hosts. It makes no sense for a virus to infect a host, cause a persistent infection, if it cannot spread to a new host, because when that host dies, the infection will be over, and that's the end of the virus. So all these persistently infected viruses, even if they're silent in the host for any period, silent meaning making no virus particles, at some point they have to make virus particles so that they can spread to a new host. And we'll see how that happens for these viruses. Now, the genome is present in all of these herpes virus persistent infected individuals, but their state differs depending on the virus. And here are the different states of the genome. One is that there is non-replicating DNA in a non-dividing cell. A non-dividing cell could be a quiescent cell or a neuron, and this is herpes simplex virus and varicella zoster virus in neurons. The cell doesn't divide, neither does the DNA. Another state is that we can have self-replicating viral DNA in a dividing cell. Epstein-Barr virus, Cytomegalovirus. Here we have a non-herpes virus, human papillomavirus, to show you that this can happen with other viruses as well. Hepatitis B virus, Kaposi's sarcoma herpes virus. And finally, the DNA can be integrated in the host chromosome and replicate with the host, and human herpes virus 6 is an example of that. So let's take a look at some of these persistent infections and see how they work. Herpes simplex virus. Over 80% of Americans are seropositive for herpes simplex viruses, and they have viral genomes in their peripheral nervous system. These are neurons outside of the brain and spinal cord. Millions of people have latent viral genomes in their peripheral nervous system, but they don't have symptoms. And as I say, all of you listening probably have herpes simplex virus DNA in your peripheral nervous system. 40 million Americans experience recurrent herpes disease, where periodically the virus will start to replicate and you have the production, say, of a cold sore or a blister. And there are two kinds of herpes simplex viruses, type 1 and type 2. Type 1 is typically limited to the head area, but not exclusively, and type 2 is sexually transmitted, but the two can be in different places as well. And this is an example of a well-adapted pathogen that mostly causes minimal disease, but establishes an infection within the host for the lifetime of the host, and also can spread to new hosts as well, as we'll see. Primary infection of herpes simplex virus. This often happens either in utero or during birth. 80% of babies are infected in this way, or you can be infected shortly after birth from your parents. Short incubation period. The infection is typically inapparent, but your baby can have fever, sore throat, lesions, etc. Then this is cleared, but the virus stays forever. So what happens is the virus infects epithelial cells, mucosal surfaces in the upper respiratory tract, typically for type 1 herpes virus. The virus replicates in these cells, can spread from cell to cell in the epithelium, but eventually viruses are shed beneath the epithelial cell layer into the subepithelial space, and this area, of course, is innervated. These viruses then infect the ends of neurons. The virus particles can spread to the cell body of the neuron, and this can be either a sensory neuron or a sympathetic neuron, and then the virus becomes latent in those neurons, as we'll see in a moment. So the primary infection early in your life leads to virus production and eventual establishment of a latent infection in neurons, peripheral neurons, not central nervous system neurons. Here's a cartoon of that whole event. Here on the left are 3 epithelial cells. The virus infects these cells, enters the end of neurons, travels to the neuron cell body, and then the DNA ends up there. So this is a single neuron in a ganglion, which is, of course, a collection of neurons. You can see the others behind it. The genome is silenced. It's coated with nucleosomes, and it is not expressed into mRNAs. Multiple copies of what we call episomal viral DNA. This is separate from the chromosome. It does not integrate into chromosomal DNA. It remains separate, and there's no replication because neurons don't divide, so there's no need for the viral DNA to divide. And unlike love, herpes is forever. Drugs and vaccines cannot cure a latent infection because they cannot get rid of this viral DNA in the neuron. If you look online, there are many people who claim that Dr. So-and-so cured their herpes infection, but that's not true. It's just not possible. In that neuron, in that latently infected neuron, the only thing that is produced is a RNA called the latency associated transcript. And this seems to be processed into microRNAs that are probably responsible for, in part responsible for maintaining latency. No proteins are made from these latency-associated transcripts. And no other RNA is made, so no proteins are made, only microRNAs. And as I said, these microRNAs probably are needed to maintain the genome in its latent state, and there are also contributions of the host as well that likely contribute to silencing. So here is the classic latent infection. This genome is in the neuron. It's almost completely silent, so it's undetectable by the immune system. Periodically, the genome undergoes what we call reactivation. And what that means is that new virus particles are made. A small number of neurons in each ganglia reactivate. The genome begins to be transcribed. Virus particles are assembled. They travel by anterograde transport back to the nerve terminus. They are released. They infect the epithelial cells. You may get a cold sore or fever blister. So this is the tissue innervated by this particular neuron. The virus will enter these cells and cause a cold sore. But not always. Sometimes you can have reactivation and virus production in the absence of having a cold sore, and that's a good way to spread it to other people because you have no idea that you're making virus. So you could spread it to your child or your lover or someone else. And that's how infection is transmitted to someone else, by contact, close contact. And it's brilliant because that's how the virus gets to another person. It's not really making you very sick. You could have a cold sore or nothing. So it's a really well-adapted pathogen, and that's how it spreads to another host, which is absolutely essential. If this latent infection stayed only in you, that would be the end of the virus. The immune response eventually kicks in and probably participates in turning off the infection, but the latently infected neurons still remain. But the response is too slow to prevent shedding, and that's because the virus encodes gene products that antagonize clearance of the virus infection. So that's an important part of being able to spread as well. Now, this whole process of reactivation, in some people happens every 2 weeks, every month they have cold sores or at least virus shedding. And in other people, they never have cold sores, although they may periodically shed virus. And some people seem to very rarely shed virus. And obviously, this is some function of the host that we haven't sorted out. Here's a picture to show you where herpes simplex type 1 virus becomes latent. in the trigeminal ganglia. And this is a ganglion, which is a collection of neurons that are tied to nerves that innervate the facial area, as you can see. So you can imagine getting an initial infection in the nasopharyngeal mucosa. It's innervated by these nerves. The virus enters these nerves, goes to the trigeminal ganglion where it becomes latent, and that's where the viral DNA is. And next time you have your hand on the side of your head, it's just above herpes simplex virus DNA infected trigeminal ganglion neurons. Then on reactivation, the virus is produced in the nerve cell, travels down the nerve, and you can see these lower nerves innervate the lip area, and that's why you get a cold sore. Makes perfect sense. Similar things happen when you're infected with genital herpes. Herpes simplex type 2, there are similar nerves and ganglia in the genital area that are infected, become latent, and also reactivate to cause cold sores as well. Now, what is this reactivation? A very carefully studied process. We'd like to know what turns on viral replication and virus production. Certain things like a lot of sun. If you go to the beach or skiing, this can often result in a cold sore. So we think UV light is participating. Stress of some kind, physical or emotional stress, can often lead to reactivation. Nerve damage, hormonal imbalance, steroids, a number of different conditions can lead to virus replication and shedding. But the common thing among all of them is that the stresses have to stimulate the production of viral proteins to activate the transcription. Remember from some time ago, here's the herpes virus genome. There is a cascade of transcriptional events where we first have immediate early genes transcribed, and then early and then late genes. And the key one is VP16, a protein needed for activation of immediate early promoters. These promoters are not recognized by host Pol2 unless VP16 is present. So somehow all these proteins have to be made to get this transcriptional program going. And recently there have been some very interesting research programs which are illuminating what's going on during reactivation. I want to tell you about the results of some of them. This is a study that has been done in mice. And as I told you earlier, you have to take it with appropriate grains of salt because we don't know if this exactly happens in people. But what we think is happening is that here we have at the top a ganglion with a neuron in which we have latent viral DNA in it. And what has been recognized is that this viral DNA is chromatinized. It's wrapped around nucleosomes. These two spheres are nucleosomes, and the black line is the viral DNA wrapped around it. And this DNA is transcriptionally silenced by histone modifications, methylations shown here on specific residues, so that the promoters for lytic replication are silenced because of this epigenetic modification. Upon stress, a particular type of stress that they use leads to activation of protein kinases. And that leads to phosphorylation of this particular histone. It's a histone H3 serine number 10, which is right next to a specific methylation mark, this one right here. And this is called a methyl phospho switch. Adding a phosphate, which is in blue here, next to this particular methylation, it displaces the repressive factors. It makes the chromatin permissive for simultaneous transcription of viral immediate early and late gene mRNAs. This is called phase 1 animation. So you're now making all of the mRNAs. Then among those are VP16. This is made during phase 1. This can associate with viral immediate early promoters, and it further activates transcription by removing the other modifications, the other methylations, causing acetylation of the chromatin, which opens it up, and now we have full reactivation. All the gene products are made. So we have a great molecular description of how stress leads to the activation of a protein kinase, a neuronal protein kinase in this way, which starts to remove the epigenetic modifications that are silencing the genome, let it be expressed. And of course, that key protein, VP16, is made so that you can turn on all the lytic gene expression. And then you have the replication cycle preceding production of infectious virus. Our next question is, persistence of herpes simplex virus in nerve ganglia requires which of the following? Continuous episomal DNA replication, low-level production of virions, silencing of all gene expression except Latent microRNA, UV light, stress, or steroids, or all of the above. If you want to think about this, pause your video now because I'm going to give you the answer. The answer is silencing of all gene expression except Lat and microRNA. We certainly do not have episomal DNA replication. Remember, the neuron's not replicating, neither is the viral DNA. There is not low-level production of virus particles. There's no virus made. And UV light stress steroids is needed for reactivation, but not for persistence. So that's why the answer is Latent and microRNA is the only things that are made from that genome in the neuron. The next herpes virus I want to consider is Epstein-Barr virus, another herpes virus that infects most adults. 95% of U.S. adults are seropositive and carry the Epstein-Barr virus genome. And they carry it in B cells, B lymphocytes. Most of you are infected at an early age and you are asymptomatic, but infection can cause mononucleosis. This infection can lead to a number of cancers. Primary infection, the virus infects mucosal epithelial cells. This is the oropharynx in this case. We have saliva, epithelium, so this is a virus transmitted commonly by saliva, by close contact. The virus infects the epithelium and then infects B cells. It can infect B cells here in the subepithelial tissues. The virus can then enter the blood in those cells. This infection can be controlled eventually by NK cells and cytotoxic T cells, but a fraction of B cells are going to remain persistently infection, and no virus proteins are going to be produced. For example, latently infected resting memory B cells. And in those resting B cells, the virus will persist. If reactivation occurs, infectious viruses will be produced. These will be controlled by cytotoxic T cells, but it will replicate sufficiently to spread to another individual. Here we have a B cell that is now lytically infected. It's been reactivated. It's shedding virus particles, which will replicate through the epithelium and be shed in the saliva. So that's how you spread the infection to a new individual. So B cells are essential for latency. They are the site of replication and latency. They persist in the host, the latently infected B cells. From time to time, they become reactivated and shed virus particles, which transmit the infection to a new host. In these latent infections, the viral DNA is a self-replicating episome in B cells. It's a little different from herpes simplex virus because it's self-replicating, because it's in B cells, and it needs to replicate with the B cells. And in a latent cell, which doesn't replicate very often, the virus makes very few copies of DNA, but it does make a limited repertoire of viral gene products. These B cells, by the way, these latently infected B cells can exist in the bone marrow and in lymphoid organs. And then periodically they become reactivated. The mechanism is not as well studied as for herpes simplex virus. The genome then begins to replicate at a higher rate to produce virus particles. And at that point, these reactivated cells are killed by CTLs because they're making viral proteins that are recognized. Latently infected B cells are not eliminated by CTLs because immune modulation by viral proteins prevents them from being targets. Next virus, herpes virus, is varicella zoster virus. This, at one time, used to be a very common cause of chickenpox, a childhood disease characterized by a skin rash. When I was a kid, everybody got chickenpox early in their lives because there was no vaccine. Now a vaccine prevents all of that. This is a virus spread by respiratory secretions. It infects your upper respiratory tract, replicates in lymph nodes, establishes a primary viremia. That brings virus to other organs, including liver, spleen, and they produce virus, giving rise to a secondary viremia. Sound familiar so far? Measles is very much like this. The secondary viremia brings virus to the skin, and you get the rash shown on the upper left here, typical of chickenpox. It's about 2 weeks after the initial infection. And then the virus enters sensory neurons that innervate the skin, and the virus establishes a latent infection in ganglia, in neurons of ganglia that are from these sensory neurons in your skin. And there the virus remains latently for many, many years, and it can be reactivated later on. Typically when you're 50 years or older, some of these initially infected neurons will start to produce virus. It will travel back to the skin and you get a skin rash, and that is called herpes zoster or shingles. And it typically is a series of rashes along a dermatome, a single area of your skin innervated from a particular ganglia. And as I said, this often happens later in life unless you are immunosuppressed, in which case it can happen at a younger age. Before the vaccine, almost everyone was infected, and about 30% of them developed zoster. Most of them, as I said, over 50 years of age. The latency is characterized by episomal viral DNA, which is at a level of 2 to 9 genomes in 1 to 7% of the neurons in the ganglia. This is non-replicating episomal DNA. There is some viral gene expression. There are some immediate, early, and late genes. Again, it's different from herpes simplex virus, which is completely silent except for the LATs. What triggers reactivation to cause the shingles are unknown. We don't have a nice model for studying this as we do for herpes simplex virus. Cytomegalovirus is another herpes virus that infects many people. Seroprevalence studies show 50 to 99% of humans throughout the world have been infected. Also transmitted by respiratory routes. There is virus in saliva. It's also present in urine, can be spread that way. It can also be sexually transmitted. This virus replicates in peripheral blood, white blood cells, and endothelial cells. This graph at the bottom is an indication of seroprevalence, cytomegalovirus IgG seroprevalence in different age groups, in different populations, non-Hispanic white, non-Hispanic black, and Mexican American. And you can see, 40 to 50% seroprevalence early in life, which increases with age. 75 plus, almost everyone is infected. The primary infection is usually asymptomatic, or maybe a febrile mono-like an illness, mono meaning mononucleosis-like illness caused by Epstein-Barr virus. We can persistently shed virus in saliva and urine for months to years. So here a little different in that we can be making virus for a long period of time. This infection is eventually resolved by cellular responses, but infected myeloid cells remain in the bone marrow. Here's the common myeloid progenitor here, which is a precursor of monocytes and macrophages and dendritic cells. These are latently infected. They remain in the bone marrow, and periodically they can reactivate, and you can shed virus, which again will spread it to other people. Now, human cytomegalovirus is a major problem for two reasons. First of all, for organ transplantation. Everyone has this infection, so if you get an organ transplant and you get immunosuppressive drugs, this virus can start replicating to very high levels and cause pathogenesis. And so that is something that transplant doctors have to pay attention to. And the second is that the virus can cross the placenta and cause severe multi-organ congenital defects and death. So again, in an infected mother, the potential is that the virus can infect the baby. And here's a figure showing the numbers here. Out of 1,000 pregnancies that lead to live birth, we have 600 women who have CMV before pregnancy, so they've been infected earlier in their life. Most of those give birth to CMV negative babies, but 6 give birth to CMV positive babies. In other words, the virus crosses the placenta, infects the baby, and a fraction of those can have permanent congenital defects. If you look at the women who do not have CMV before pregnancy, 7 of them will get CMV. Most of them do not get CMV during pregnancy, but some of them will. And of those, 5 give birth to CMV negative babies, and some will have positive. So 1 to 2 babies out of 1,000 pregnancies have issues because of CMV-induced congenital birth defects. And so that's a big problem. And when Zika virus emerged a couple of years ago, And the world learned that this virus could cause congenital birth defects as well by crossing the placenta in an infected mother. People didn't realize that CMV has already been known for years to cause similar problems. I recall speaking with journalists about the Zika problem and telling them, there are other viruses that cause congenital birth defects like human cytomegalovirus, rubella virus. And I was amazed that few journalists recognized this. So Zika virus congenital birth defects is not new among viruses. Our last question is, what do persistent infections with Epstein-Barr virus, varicella-zoster virus, and cytomegalovirus have in common? B cells are essential for latent infection. They may cause congenital birth defects. Viral DNA persists as an episome. The factors governing reactivation are well known. The answer is C. Viral DNA persists as an episome. B cells are only essential for EBV latent infection. Only CMV is known to cause congenital birth defects, and certainly the factors governing reactivation are not well known. So that leaves us with DNA as an episome, which is true, and that's what I've told you for all these viruses. Our last herpes viruses are human herpes viruses 6 and 7. After the herpes viruses that I've told you, these are named by numbers. These are agents of exanthem subitum, which is a mild childhood rash. You can see a child here with a rash typical of these viruses. This rash is called sixth disease. It's the sixth of all the childhood rash diseases. Many adults are infected, and the viruses are passed from parent to child in respiratory secretions. The viruses infect lymphoid cells, endothelial cells, liver cells, even the CNS and salivary cells. And as far as we understand, for HHV6, latency is in monocytes and macrophages and hematopoietic precursors, and 7 in CD4 positive lymphocytes. So these are viruses that become latent in cells of the immune system. Herpes 6 integration has been found to occur in a small fraction of individuals, 1%. The viral DNA integrates into telomeres. This is very unusual. So here in this diagram is a human chromosome at the top in blue and the telomeric region at the end, shown in red. These are the highly repeated regions. The viral genome of HHV6 is shown in the middle. You can see direct repeats at either end, and telomeric repeats, similar to those in the human chromosome, are shown in red. And at the bottom is an integrated copy of HHV6 at the end of a human chromosome, and it's integrated into the telomere of the chromosome. This is not common. It only happens about 1% of the time, but this would allow the virus to be transmitted through the germline because it ends up being in the germline of people who are infected. So this is one strategy for latency and transmission, that the virus genome integrates into the human chromosome, which is not the case for the other herpes viruses that we've talked about. And of course, the transmission would be in these individuals, by the germline. In the other 99%, the transmission is by infection. But this is a specific case, a special case where genome integration can allow transmission. Let me end up with this amazing slide, which shows the number of humans infected with different chronic viruses. And this is estimated from the percentage of humans carrying a given virus, assuming that the world population is about 7 billion people. And this is seroprevalence data, where you look at sera and say who has antibodies to which virus. And this is typically only obtained from developed countries. So the impact of chronic infections on humanity as a whole is really not well characterized. A rough calculation shows that there are many billions of chronic infections in humans, with each of the 7 billion of us harboring somewhere between 8 and 12 chronic infections. And that's what we know about. Remember, that's what I told you at the very beginning in the first lecture. I said we all have about a dozen herpes viruses in us, and of course many, many more, because we're not looking for them by seroprevalence studies. So here we can see all the different viruses on the x-axis, estimated number of infected individuals on the y-axis. And endogenous retroviruses, ERVs, those are present in everyone. So that represents 100% of the world population, whatever it is, 7 billion or so, when this study was done. And then you have anelloviruses. These are small single-stranded DNA viruses that infect almost everyone. We don't know what they do. Human herpes 6 and 7, varicella-zoster virus, Epstein-Barr, Cytomegalovirus, polyomaviruses, adeno-associated virus, herpes simplex viruses. Now we have a decrease, fewer people infected with hepatitis B and C viruses, adenoviruses, HIV. The world burden is 35 million or so of HIV. This is amazing that so many people have, and these are just the viruses we're looking for. Of course, there are plenty more in us that we don't even look for. So what's the point of this? These viruses are so common that it would be really hard to find uninfected control parents. If you're doing a study to identify viral associations with disease or effects on the immune system. If you're studying a particular virus and you want to know its effect, those people are all infected with these and more viruses. So how are you going to do that study? You don't know the contribution of these viruses. The fact that most of us do not show symptoms of infections by these viruses on this chart make them part of the metagenome of normal individuals. So we need to really think about what these infections mean for humans without disease and the need to understand chronic diseases that are considered non-viral based on our ability to compare infected to non-infected individuals. So just think of all the chronic diseases that we study that are not viral, at least we don't think they are, and we're trying to figure out what's causing them. Take cancers or diabetes or high blood pressure or heart disease. How can you understand those diseases when there's no one who is uninfected, as you can see from this chart? Next time, we're going to consider a certain type of persistent infection that can lead to cancer.